we go. All right. Pleased to introduce our next speaker, William Neirman, who's Director of Infectious Disease, the Infectious Disease Program at the J. Craig Venter Institute. Dr. Neirman conducts studies of human respiratory system, microbiome of infants in South Africa and in the Philippines. He's going to present on the microbiome and our genome. Sounds like a lot in 20 minutes.
Thank you very much, Leanne. I'd like to thank the organizers for inviting Karen Nelson to present today. She sent her regrets, and because I'm not Karen, as you note. But by way of a little bit of defense, basically JCVI, the Venter Institute, did some really pioneering work in the early days of microbiome analysis. It started out with Craig sending his sailing yacht around the world a couple times, sampling the surface waters, really flooding GenBank with genes and proteins and organisms at an incredible rate. And at the same time, they started with Karen Nelson a project that none of us were particularly enthused about, that was referred to as the POOP project, so you can imagine what that was. And with that, let me begin this overview of the human microbiome. So the human microbiome, I guess the recognition of its presence and significance started to emerge somewhere around 2000. The numbers are highly speculative here in terms of numbers of bacterial cells compared to human cells. And of note, I think, in particular, because I think it's very important for microbiome issues, is the numbers of genes that are encoded in the microbiome on our bodies. Wildly estimated to be 150 times the numbers of human genes. One thing that's of note for those of us at genome institutes that have been doing microbial genomics for a long time is it's really a striking feature. Well, there's two striking features of the microbiome. Number one is that probably half of those microbial genes in our bodies are totally function unknown. And the other aspect of that is the vast majority of these organisms have never been cultured. So we've never looked at them in the way we traditionally study bacteria. Through cultivation and really reductionist analysis of how those bacteria do their things. So these are actually bacteria, fungi, and viruses. It's estimated that the microbial component of our bodies encompasses about 3 pounds. It's really clearly established that it makes major contributions to our metabolism, perhaps in very many essential ways. One of the early things that I was struck by in discussions with a collaborator from my fungal background was the suggestion I made, well, let's use for what we want to do these gnotobiotic mice, these mice that are germ-free. And the response came back, those mice are really anything but normal. They don't have the ability to develop normal immune systems, and they have all kinds of peculiarities simply because they've never been exposed to microbes. So I think the last line here is really a true statement. Any aspect you might consider the human microbiome is really poorly understood. We acquire our microbiomes at birth. I've got a project that was mentioned where we're sampling human infants from age 0, actually sampling the first time on their birthday, maybe a few hours after they're born. And now we've taken that out for 2 years. Here, it shows how the infants pick up microbiomes from their mothers. Our focus, of course, is on the respiratory tract microbiome, but the respiratory tract actually ends up picking things up from everywhere. You see in number 2 here that this is written, somebody asked me, Meredith, who's going to follow me, asked me if I was going to talk about the gut microbiome. The fact of the matter is that most of the work that's out there is the gut microbiome. I'm fairly novel in that I've never been particularly interested in going there. I like the respiratory system. But apparently the gut microbiome, which approaches an adult status in close to 1 to 2 years, in the respiratory tract, our infants at 2 years, we sample them every month and we see wild variation month to month, even 2 years out. So this is going to be site-specific and microbiome-specific and probably environment-specific as well in terms of when the respiratory tract stabilizes. And I think we don't know anything about stabilizing the other components of our bodies where the microbiome exists. I alluded to the role microbiome plays in immune development in animal models for sure, but I think it's clear in humans as well. I think there's correlations with lots of these things, especially with immune-mediated diseases. Certainly antibiotic usage, one of the issues that we were talking about when we were talking about mice is humans harbor Candida albicans. It's the fungus that causes thrush, and Candida albicans is part of our commensal microbiome. It's there all the time. Mice do not harbor Candida, but you can manipulate mice by antibiotic treatments where the Candida is just as happy in mice as they are in people. So this is really mediated by modulating the microbiome in the mouse. And so it's an example of the potential of modulating microbiomes to achieve some therapeutic or other interventionist effect, but that's just one of the promises that's still in the microbiome issues. Certainly, microbiome differences have been observed in weight BMIs relative to the discussion at this meeting. Diet is very dramatic in its modulation of the gut microbiome, and certainly health status. I think it's generally true everywhere, whatever you look at in disease versus health, that the microbiome associated with the site of a particular disease, and of course my perspective is infectious diseases, is characterized by really a collapse in the diversity of the microbiome and the dominance in infectious diseases of one or a few pathogens. The microbiome is really, as you might expect, site-specific. The respiratory tract microbiome is totally different than the gut, and it's totally different than the skin. This is just like if you grew things in a tropical rainforest in the soil or in a desert, you expect it's different because the eco-niche is quite different, and the microbes adapt to our bodies in the same way. Certainly there's we all like to think in infectious diseases that there's a healthy microbiome that's protective for infectious diseases. And as a consequence of things that contribute to alterations in the microbiome as well as other aspects of the host environment, a condition of dysbiosis sets in that predisposes us for a lot of problems: enhanced inflammation, permeability in the guts, transfer in the severe cases, for example, in non-alcoholic liver fatty accumulation disease. You actually get bacteria leaking through the gut wall and being transported directly to the liver, as well as all the metabolites that are being produced by the gut microbiome. So with the Global Ocean Sampling Project in the databases and a couple of key papers put out by Tiger, the Institute for Genomic Research, as we were called, and other places, we actually did in 2005, 2006, from what I can tell in searching the literature, the first 16S profile, which is 16S ribosomal profiling to identify what bacteria or archaea are there in the relative abundance, and the whole microbiome metagenome sequencing approach, which gives you more information at a much greater cost. Papers were first published in 2006 on these. So the NIH Roadmap established the Human Microbiome Program in the US. There were subsequently in several places in the world similar kinds of undertakings to explore the human microbiome from the perspective of really developing an awareness and some tools for the microbiome and what's there in healthy individuals. So this was conducted in the U.S. and the Human Microbiome Program at Boston, at the Broad Institute, Washington University, our institute, JCVI, and at Baylor, as 4 large-scale sequencing centers coordinated through NIH, multiple institutes in a very controlled environment where they spent days each week on the phone talking to each other to establish protocols, establish SOP standards. The goal of the project was to look in healthy individuals, several hundred healthy individuals at selected sites, and to, in parallel, which is really essential for doing the kinds of microbiome work we need to do, given that not much was known about the organisms in the microbiome at this point, to determine whole genome sequencing of a whole bunch of reference organisms that are representative of the components of the human microbiome. So this is a breakout of the sites that were the target of the NIH Human Microbiome Project. Multiple sites in the oral cavity, a couple sites on the skin. They did nasal as well, of course, the gut microbiome, and a couple of sites within the, to explore the vaginal microbiome. As I already alluded, each site was different. There's only thing that you see in terms of an overlap is the external nares sites with the skin, which, the external nares is just one minor step away from the skin. So that's not surprising that the eco-niche there is much different. Everything else is totally distinct. One of the things that's come out of this is, well, besides the fact that there's no apparent relationship with gender, age, weight, ethnicity, or race, there's also tremendous individual variability at the same site between normal individuals. So microbiome data sets are always very noisy data sets. You need a lot of controls and appropriate study design to have power to really form associations and come up with biologically relevant conclusions for microbiome studies. A striking thing about the oral cavity, where there were multiple sites sampled, is there's dramatic differences between sites that are very close, such as the dental plaque versus gums. I guess it's not surprising, but there is that kind of diversity that exists in regions of the body that are even very close proximity. One thing I think that's really becoming obvious in microbiome studies is when you do 16S profiling, you get a view of who's there in relative abundance at the genus level. But digging into the genus level, you find you have basically no information on species to go there. You're going to have to do either specific PCR assays, or you're going to have to do whole microbiome sequencing, metagenomic sequencing. So at the 16S level, the listing of who's there from a genus level is potentially not all that informative, because very important functional differences exist between species within any genus, or even within strains within a species, as I'll show you an example in just a little bit. So the example here that's offered here is the lab strain E. coli of E. coli K12 with its metabolic capabilities, and the E. coli O157H7 highly virulent pathogen. E. coli in particular has a very variable genome with very variable metabolic capabilities. And many of the phenotypic associations with microbiome are being teased out at the level of the metabolism of the microbiome that's present and the metabolic products that are being produced by the microbiome. On the other side of the issue is certainly the microbiome provides many metabolic capabilities that supplement our human human encoded metabolic capabilities for example, digesting complex carbohydrates and such. There's lots of associations with microbiome and diseases. The details are the topics of study across all of these, and at JCVI and probably lots of other places, there's projects devoted to focusing on many of these kinds of associations that have been revealed. And again, I'm alluding to almost everything here is from studies associated with the gut microbiome. This is just a quick slide on the Cancer Association. Of course, Helicobacter pylori came out a long time ago. That's not colon, of course. That's stomach cancer. To establish, and Helicobacter pylori is really kept in check with a healthy stomach microbiome. And the other example, Clostridium difficile, in the lower digestive tract is kept in check with a healthy microbiome. But for the cancer perspective, it's been clearly established that diet is really important to cancer incidence. I was at an aging microbiome meeting, and it was cited, woo. Anyway, what you eat is turned into metabolites by the organisms, and the metabolites have consequences for predispositions to colon cancer, such that in the rural diet in South Africa versus a Western diet in Southern California, there's normal incidence of colon cancer versus no colon cancer. And people have actually established associations of the bacterial metabolites that are contributing to these dramatically different outcomes for these diseases. So I'm going to skip some of these things. I can't help but spend a minute on this because everybody, I'm sure, has heard of fecal transplants and the therapeutic efficacy of doing this. This paper came out in 2013. Sarah Highlander at our institute provided us all with this slide, pointing out that 2013 was not the first time this came about. This was a paper reported in 1989 using a select set of identified strains of these organisms, and Sarah was actually working on coming up with an intervention of pure cultures, mostly lactobacilli, and it was her point that it's the lactobacillus whatever species strain that was essential for suppressing the C. diff overgrowth in the gut and causing remission of symptoms very quickly. Infectious diseases, I should just mention, one of the things that are particularly of interest to me is the fact that when you have a severe flu, the flu virus modulates the immune system of the host. It results in a high incidence of strep pneumonias following the flu. And so all of these things are hooked together with the microbiome, the host immune status, and the pathogens that are actually already in our respiratory tract microbiome. They're all there. I don't think we catch bacterial infections in our lungs. I think we may catch flu, but a lot of things happen that bring out these things like strep pneumo that are commensals in our nose and commensals in our respiratory tract to cause the severe respiratory infections. We all want to look to interventions. The potential is there for microbiome to actually bring these things about. There's a lot of reservations within the community at large that the microbiome has been oversold. I suspect in the early days of epigenetic marking of the genome, there was a lot of criticisms raised to that as well. And undoubtedly, it has been oversold to some extent in particular circumstances. There's a lot of them there. But to go to the focus of the meeting, a lot of data associating microbiome with obesity have been done in animal models. and now that the numbers of studies have accumulated, it's not clear that there's an answer. There's certainly no characteristic microbiome components for the human that can be shown to be associated with obesity, and this is a 2014 review. It's generally true that obese individuals harbor a less divisive bacterial communities. This is the same situation as I told you for respiratory tract infections. When things are bad, the microbiome diversity goes down. Certainly, there's mechanisms, undoubtedly not demonstrated yet, where the microbiome will influence BMI, and potentially therapeutics can be developed for this. So, now just to get into a broader philosophy on microbiome and everything, this is a what's really, I think, a current paradigm in infectious diseases, where it's not the microbiome, it's not the pathogens, it's not the host, it's everything together, and all of these qualifiers on all of these three arms of this triangle that come together to have you experience a severe infectious invasive infection. And so I quickly put together the obesity-host-microbiome-innate system paradigm, completely fantasy at this moment. Obesity, undoubtedly, it wouldn't be restricted to obesity, metabolic syndrome, any autoimmune diseases. All of these things probably are a complex interplay of host genetics, but maybe not even just host genetics, because now, of course, now that I've been to this meeting, it's host genetics, including the epigenetic marks in the host genome. The innate immune system status is really important in infectious diseases, and it's important in the developing of a lot of things associated with the gut microbiome, like colon cancer and other things. And the third component here, the microbiome metabolic function, undoubtedly is where the action is. It's not in the various ratios of the various genuses present. It's in their metabolic capabilities and what they're actually doing in the gut to either facilitate or or destroy homeostasis in the individual. So I think the way to approach this in the future, and it's currently the model for the Human Longevity Incorporated, Craig Venter's new for-profit, latest for-profit undertaking, is really looking at putting everything together that's going to play the genome, the very standardized patient phenotyping, the microbiome, and really in a very long-term project, put all these things in such a way that you can actually tease out all the details that are associated with the kinds of interactions we're talking about at this meeting, whether it's infectious diseases or obesity and how it's modulated. Lots of participants and funding organizations. With that, I'll quit. Thank you.